Thank you so much. I'm so glad to be here, and also I'm glad to see so many chewers here in the audience. My name is Trulsson, and I'm a professor in oral rehabilitation at the Department of Dental Medicine at Karolinska Institutet. Normally, I have my basic training, I can say, in neuroscience, which is a bit odd for a dentist, but I'm also training clinically in oral prosthodontics. Normally, I talk about masticatory function, how the brain controls masticatory function, and what happens with masticatory function during oral rehabilitation procedures. But I will not talk about that today. I will talk about, is chewing really important? Why are we chewing? And is it good for us to chew? And I will also show you how you can chew your way to a better brain. So, chew before you swallow. It's a famous words said by a famous person. Do anyone know who it is? It's not Hippocrates. He could have said it, but it's not him. And it's not a tweet by Donald Trump, I'm sure about that. It's actually this guy, George W. Bush. 2002, he was president of the United States of America. He was sitting in the White House watching a football game, and he got a pretzel in his throat. He choked, he fainted actually, and fell on the floor and hurt himself quite badly. So a couple of days later, he had this press conference, and he told the journalists that my mother always told me, chew before you swallow. You should listen to your mothers, he said. So we can learn two things from this. You should always listen to your mothers. That's always the right thing to do. But it's also, we also learn that it's important to chew before you swallow. If you don't do that, you can hurt yourself and you can even die. And that brings us to the subject for today. Why is chewing important? The first thing we think about chewing is, of course, that it's a mechanical breakdown of food. That's the important thing. And that means that we decrease particle size of the food. If we don't do that, we can't swallow properly. While doing that, we also increase the surface area of the food, which is important when the food comes down in the gut, and the bacteria and enzymes will take care of it. So, of course, that is important. While chewing, we also increase the salivation. It increases with about 100%, actually, when we chew. And that is mostly because of the parotid glands that produce a very watery saliva, also full of enzymes. And this is important for preparing the bolus for swallowing. The bolus, that is the food in the mouth while chewing. It's called bolus. And when we decide to swallow, it's quite difficult to say why we decide to swallow, but it's quite a lot of criteria that has to be fulfilled to swallow the bolus. It has to be the right mechanical properties, it has to be the right particle size, it has to be the right temperature. And then we can swallow the bolus safely. So chewing is, of course, very important to form this bolus in the right way. The first stage of the chemical digestion is also happening inside the mouth during chewing. Through breakdown of starch, through amylase. Amylase is secreted in the parotid glands and is in the saliva, and it's mashed into the food, and it starts the breakdown of starch in the bolus, already in the mouth. And it's also going down in the stomach, and is also working up to half an hour in the stomach. Chewing also triggers reflexes that prepare the digestive tract for food. And this is quite interesting. I mean, you all have chewed chewing gum sometimes, and maybe you have felt that you get quite a lot of acid in the stomach. This is a very typical reflex that is elicited, is triggered from chewing. And we have like hundreds of these type of reflexes. And to be honest, we don't really know so much about their importance. I think that in the coming years, we will be much better at this and really understand the importance of chewing for these type of reflexes and how it affects our digestion of food. Being able to eat a nutritious meal in a social context. If we can't chew properly, and maybe we have like false teeth that move around when we eat, it's very difficult to eat nutritious food. Nutritious food tend to be kind of hard, like fresh vegetables, fresh fruit, and nuts and stuff like that. And that's very difficult to eat if you can't really chew well. So then you tend to put that aside and choose other food that is really mushy or already chewed from beginning, and that is not really the most healthy food. In a social context, that is also very important. It is important for us to chew well to be able to be in a social context. If we can't really chew well and we lose our false teeth during the dinner, we decide not to have dinner with our friends anymore. It's socially not acceptable. So then you get isolated, and we all know that isolation, social isolation, is not a very good thing for healthy aging. Release of substances in the brain like serotonin that will affect mood and also coping with stress. Professor Per Södersten is a colleague of mine here at Karolinska Institutet. He has his office not so far away from here. He used to say that you are not what you eat, you are how you eat. Professor Per Södersten and his colleagues, they are doing research, and they also have a clinic working with patients with eating disorders. Patients with anorexia, they stop eating. They also stop chewing. And one part of their treatment is to use this mandometer to track their behavior and also change their eating behavior. So they start to eat and chew in a normal pattern. And this makes them feel better. The theory behind this is that we have the chewing center in the brainstem, which affects the raphe nucleus in the brainstem that elicits serotonin to the prefrontal cortex, and you actually feel better. You increase the mood. So while they actually get the patients to eat in a normal way and increase their chewing, they also feel better. So, why do we like to chew chewing gum? I think that's a fair question. I mean, chewing gum, it doesn't taste so good. It tastes good in the beginning, but after a while it doesn't really taste anymore. So why are we chewing gum? Must be a reason for it. There has been quite a lot of psychological studies on this topic. And one of the most interesting things is actually that if you take a group of students like you and put you in a stressful situation, like an intelligence test, you will see that if you chew chewing gum during the same time, you will get a better result. You will get more intelligent. Of course, you don't really get more intelligent of chewing gum. You don't even get a better memory. But what happens is this. Chewing gum generates improved attention, improved alertness. You actually get a bit more awake when you chew. It's elevated mood. You feel a bit better when you're chewing. And lower perceived stress. It's like a stress coping behavior to chew. I mean, you just have to look at the hockey coaches to understand why they are chewing a lot. Of course, they are stressed. So you may actually say that chewing gum is a functional food without food. So let's go now to the last puzzle, the piece of the puzzle here. Why chewing is important. Chewing affects the brain's cognitive mechanisms. This is a quite weird statement, isn't it? Could this really be true? Do anyone know who this is? I think someone would know who this is. This is like a superhero in dentistry, I can tell you. This is Professor Per-Ingvar Brånemark. Unfortunately, he died a couple of years ago. This is the man who invented the modern dental implant. And he took a basic science finding to a multi-million-dollar industry, and also he has helped millions and millions of people in the world that has chewing problems. He's a superman. The thing is that 2001, he became honorary doctor here at Karolinska. And then he was here, right in this house here. And he had a lecture. And of course, I went to this lecture and listened to him. And then he showed this picture here. First he said, look at this picture, he said. Have you a patient with all their teeth. The brain functions well. Both the sensory part of the brain and the motor part of the brain works fantastic. And if you amputate the patient, if you take away all their teeth, you will get problems with the sensory projections to the cortex. You will get problems with the motor part of the cortex, and maybe the patient has a higher risk to be demented, to get Alzheimer. And I thought that, oh my God, what is he saying here? And then he said that if we now put implants in and can rehabilitate the chewing function, maybe we get a better sensory projections to the cortex with a lot of more activity in the sensory cortex and more motor activity in the cortex, and maybe we have a less risk of getting demented. And at that time I was a bit younger. I thought I knew something about neuroscience. I thought I knew something about dentistry, and I've never heard this before. So I was a bit intrigued here. I went down to Professor Brånemark afterwards, and I said very politely, "Dear Professor Per-Ingvar Brånemark, the things you said about cognition and chewing, I guess you haven't got any scientific evidence for that." And he just looked at me and said, "Mats, when you say something, you have to have scientific evidence for it. But I'm extremely old, and I have the right to be a visionary," he said. I couldn't say anything about that. Of course he's right. So he was a visionary. And a couple of years later, I sat down and tried to look in the literature to see if I could find any evidence for this. And I must tell you that I was very, very surprised to see that I found about 50 papers describing a possible connection between chewing and cognitive function of the brain. And I can tell you that now, if I would do the same thing now, like 15 years later, I would find maybe 1,000 papers that show this association. So I will go through parts of it with you to explain what has been shown, and then we will see if we can believe in this or not. So this is the first study by Professor Unosuka from Japan. It was published in 1999, and this is on rodents, on mice. What they did was that they studied the neurobiological effects of missing teeth in age SAMP8 mice. It's a mice that is genetically changed so that it gets old very fast. Very practical to do science on it. They cut the crowns of the maxillary molars, making it impossible for the mouse to chew normally, which led to impaired learning in its spatial memory tests and decrease of the neural density in hippocampus. So they got both a behavioral problem with that the mice couldn't find their way in a labyrinth, and also neurobiological effects in hippocampus. Here you have this water maze that they used for the mice. It's actually like a bucket of water. You put down the mice in it, and the mice is supposed to find the platform that is not visible under the surface. So it has to swim around and try to find it. When it has found it, it's of course easier next time because it can use the spatial memory to remember where the platform is. So it's like a measurement of the spatial memory, we can say. And hippocampus is a collection of cells deep inside the brain that we know is important for memory functions. So here you have the mice swimming around the first time trying to find the platform. Of course, it takes a lot of time. The 8th time it's trying to do this, it knows where the platform is, so it can find it. So look at this graph now. We have day 1 to 9. Escape latency, that's the time it takes for the mice to find the platform. This is the controls now. It takes about 60 seconds for the mice the first time to find the platform, and then it learns, and in about 6 days it's down at about 10 seconds, and then it doesn't get any better because it takes about 10 seconds to swim to the platform. So then it has learned where the platform is. 6 days. Now take a look at this molarless mice. Now they have cut off the molars, or they have taken away the molars, so that the mice can't chew anymore. They don't learn as fast. and they don't get as good either. They end here at about 20 to 25 seconds. So they don't get as smart as the ones with the molars. And this could also be correlated to number of neurons in certain parts of the hippocampus. The molarless mice had significantly lower number of neurons in certain parts of the hippocampus, which is quite scary. 2002, they published this paper where they showed that reduced chewing ability can be linked to a reduction in the number of active cells in hippocampus. The activity was measured with the FOS protein, which is like a marker of activity in the neurons. But what was very interesting with this paper is that they showed that rehabilitation of the dentition improves learning ability and increases neural activity in hippocampus. Wow, I must say. At least as a prosthodontist, this is really interesting. Look at this now. Look at the graph up here. You recognize this now? 60 seconds for the controls, the green line here. In 6 days, they are down at 10 seconds, and it takes 10 seconds to find the platform. The blue ones are the molarless mice. They don't learn as fast, and they get down to about 20 seconds. And they keep constantly now on 20 seconds. Look now on the red curve here. They are also molarless. Here they have cut off the molars. So you can see that they behave just like the blue ones here. But here at the arrow, they put back the crowns on the teeth. So they rehabilitate their teeth so they can chew again. And what happens then is that they get worse the first day, but then they constantly get better and get closer and closer to the controls that have their molar intact. Quite amazing. If you now look at the FOS positive cells and count them here, we can see a lot of active cells in hippocampus for the controls. We have much less active cells for the molarless, and for the ones that have got an oral rehabilitation, got their molars back again, you can see more active cells in hippocampus. There are a lot of these type of studies in the literature. I just picked out a few of them, but what is quite interesting is now that you can see that all measures that affect normal chewing function produce similar effects as removing or cutting the molars. Like increasing the height of the molars. If you increase the height of the molars, it will be difficult for the animals to chew properly, and that will get the same effect. You can even give them soft food instead of hard food. The same type of food, but you mash it so that it gets soft. That will also get the same effect. With problems to find their way, spatial memory is destroyed, and also neurological effects on hippocampus. So if I try to summarize now the work from the Japanese group here, we can say that disturbance of normal mastication in rodents accelerates an age-dependent impairment of learning ability combined with significant degenerative changes in the brain. It has to be aged mice. If you did this on young mice, it didn't happen anything. So it has to be an age-dependent decrease of learning ability. And the interesting thing is that in this case, we can actually talk about the causal relationship. We know that if we mess up the chewing capacity in rodents, they will get this problem with memory and also signs in the hippocampus. So what about humans now? Do you think that we can see the same thing in humans? The interesting thing is that there are a lot of papers from Japan and other places in the world, but we also have some very interesting papers from Sweden. This is a group in Umeå working with, it's under the leadership of Lars-Gunnar Nilsson. It's called the Betula study from Umeå. And they do a study to see what factors are important for healthy aging. And in this paper, they have performed clinical oral examination and cognitive tests in 273 participants in Umeå. And they found that a number of teeth was significantly associated with episodic and semantic memory. So here they can show an association between number of teeth and cognitive function. In this interesting paper from US from 2007, tooth loss, dementia, and neuropathy in the nun study. This is an excellent study. They have followed a group of Catholic nuns in Texas for decades. And then in the end, they can actually also look at their brains when they are dead. So it's a fantastic study. What they did here was the longitudinal dental records and medical examinations of 144 Milwaukee nuns. They showed that participants with the fewest teeth had the highest risk of prevalence and incidence of dementia. So here they can show on an association between number of teeth and development of dementia. We also did a study on this here. It's a collaboration between Department of Dental Medicine and Aging Research Center here at Karolinska. And here we used the SWE-OLD study. That's over 500 older people living at Kungsholmen. And here we could show that older Swedes with good chewing ability had good cognitive function. Here we could also see that it was not really the teeth, the number of teeth that was important. It was the subjective feeling of that you actually could chew hard stuff. If you could chew anything, you had a good cognitive function. What we also saw that was chewing with natural teeth or with prosthesis did not matter as long as they had no chewing problems, which is of course encouraging for dentists, because if you can give them like a prosthesis or any dental treatment that will increase their chewing capacity, it will also affect the brain. So, in humans, we can't really say that we have a causal relationship until we have done an intervention. We have to do an intervention to really see if this is true or not, because it could be some other factor that is affecting this. So now we're actually doing this. Cognitive changes and neural correlates after rehabilitation or mastication in elderly in intervention study. This is a PhD project, and the PhD student is Linn Hedberg. And this is a collaboration between Karolinska Institutet, Stockholms sjukhem, and Folktandvården in Stockholm. The aim is to evaluate if prosthetic intervention with improved muscular function has a positive effect on cognitive function and brain plasticity. So the very simple idea here is to take patients that looks like this. They definitely have chewing problems. We test their memory functions and do some brain scanning before their treatment. We treat them and so that they look like this and have a good chewing function. And then we do the memory tests afterwards and also brain scanning. Unfortunately, we don't really have any results yet. So I had to come back in about a couple of years to show you. But we have some really interesting pilot data. In this pilot study, we used verbal fluency test. In this study, in this test, you actually ask the subject to say as many animals as you can do in 1 minute. And we tested before, we have two groups here. We have a control group and we have a test group. The control group, we tested, that group was rehabilitated from the beginning, so they already had a good chewing function. So we tested them twice, just to see how a second test would affect the result. And here you can see the result for the control group at test 1 and at test 2. And of course the result on the test 2 is better, because you always get that type of effect, test-retest effect, when doing these studies. But now take a look at this test group here. They were tested before and after rehabilitation. So this was before rehabilitation. You can see that they have definitely a lower value than the control group before rehabilitation. And then after rehabilitation, we can see that they become much better, and their increase in the result is much greater than in the control group. So in the end, after the rehabilitation, they are very close in result to the control group. It was just 10 subjects in each group here, so we don't have any statistical significance on this, but it's very, very close. And it's definitely a trend that shows that it may be possible, actually, that dental rehabilitation may also affect the brain in humans. So let's get back to this visionary guy now, the Superman. Was he right or was he wrong? If you look at animal studies, we can say that we have a causal relationship. If you mess up the chewing in animals, you will get problems with memory, and you can also see effects in the brain. And if you rehabilitate them, you will see improvement of the memory function. You also see an improvement of active neurons in hippocampus. When it comes to humans, we have a lot of studies where you can see an association between number of teeth and chewing function and cognitive function and even development of dementia. But we don't really have any intervention study yet to really say that this is true also in humans. But if you ask me, I would guess that this old man, he was right. Thank you for listening.